All right, part 3 of chapter 3 is all about eukaryotic cells. So we have a lot of organelles to discuss here. This is a very generalized eukaryotic cell, but we can see a lot of membranes inside this cell. We have a nucleus with a nuclear envelope. So we're going to go through each one of these organelles. So what you want to know for this part is basically the structure of these the structure of these organelles and the structure and function. This is just showing you different kinds of shapes that we have for eukaryotic cells. We saw some of these before, but this is going to be a form of algae. Remember, algae is a plant-like protist. It's not a true plant. This is, again, one of our trypanosome. This is a protist again that is animal-like, and it can cause disease. This is causes African sleeping sickness. You don't need to know that now. These are some other guys. I'm not sure what they are. This is a paramecium, and then here we have a blood smear. These are our blood cells, and what we see in this blood cell is a little protist, and it's called Plasmodium, and it causes malaria. Vorticella, sorry. Vorticella is also a type of algae. All right, eukaryotic cells, they have a nucleus. A nucleus is where we have our DNA. It is protected by a nuclear envelope, which is actually double membrane. So the nucleus has two membranes to it. It is protected by this membrane, but molecules do need to come in and out of the nucleus. So it will have pores set up in the nuclear membrane to allow those molecules to move through. So the nuclear envelope is basically the membrane around the nucleus. So it has an envelope and then pores. Inside the nucleus, the cytoplasm just has a special name called nucleoplasm. We have chromatin, which is basically the DNA. Okay, so chromatin is a form of DNA that we have in eukaryotic cells. It is associated, remember I said prokaryotes have a naked DNA, but eukaryotes have histones around the DNA to organize it. These proteins plus the DNA make chromatin. Now, what do we have here? We have a nucleolus. Okay, so within eukaryotic cells, there's all the DNA that it requires is basically the genome. When you talk about an organism's genome, it's all found in the nucleus. So the other thing here that we see is a nucleolus. The nucleolus is a dark, dense area inside the nucleus, and it's completely different. It's where ribosomal RNA is formed. And so in the pre-ribosomal assembly. So basically those ribosomes we talked about that make protein are part of that protein, part of the ribosome, which is the RNA. That little R here stands for ribosomal, is in the nucleus, is made in the nucleus. So here's a nice picture of our nuclear envelope, and then we have a pore that's barely seen. And then we have our, the DNA, the chromatin here, and then we have our dark staining nucleolus. Now, the endomembrane system is all of the membranes inside the cell that either physically contact each other or they share membranes by budding off vesicles and then fusing those vesicles. So let's talk about what's happening here. They're going to facilitate movement of materials through the cell and also out the cell. What we have looking at here, we have one organelle called the rough ER and another very similar organelle called the smooth ER. So ER stands for endoplasmic reticulum. And let me see if I have other images of this. Nope. Okay, so rough ER, why is it rough? What are those red dots? Those red dots are ribosomes, okay? Ribosomes, I'll just put ribo. Ribosomes are embedded within the rough ER, and since ribosomes make protein, the rough ER is busy making protein, But the proteins that are made here are usually secreted from the cell. So remember, ribosomes also exist outside the cell. Ribosomes are like an upside down snowman. They have 2 subunits. Those ribosomes that are in the cytoplasm make protein, but the protein usually stays inside the cell and kind of, works in the cell like an enzyme. And then the proteins made within the membrane will be carried out of the cell. Okay, so that's the rough ER. The smooth ER does not have those ribosomes, so it's not functioning to make protein. It's actually an area where we can make fats. So lipid synthesis is happening in the smooth ER. And other functions like detoxification, this can take molecules that may be harmful to the cell, right, and change them to a form that's more water soluble, and then you can urinate it out. It can be excreted through the urinary system for us. Then we have, so that's basically it. Smooth ER is a lipid making, so phospholipids for the membrane are made here, and also detoxification. What we see next in this picture is something called a vesicle, which is how cells carry cargo around inside. So if a protein was made here, the membrane would form an outpocketing, right? So the membrane would do this. And then the newly made proteins would be stuck inside. And then eventually this outpocketing would bud off, right, to form a free membrane-bound structure, which is called a vesicle. And these guys are moved around the cell. They don't float. They just move. They're physically carried from one area to another by the cytoskeleton. And vesicles in this picture shows basically if you have a protein made here, it'll bud off into a vesicle. This membrane is going to fuse with the membrane of the Golgi apparatus. The Golgi apparatus is, the structure of it is basically like pita bread, like maybe 5 or 7 pita breads. So it's hollow on the inside and it's membrane on the outside. What the Golgi apparatus does is it can modify, just put mod, it can modify those proteins coming from the ER, the rough ER, or it can modify fats. But the Golgi apparatus is kind of a crazy, unique organelle. Not only does it modify the proteins, and what is modify, right? So maybe a protein needs a sugar group, right? Like those glycoproteins on the surface of the membrane. That happens in the Golgi apparatus. Maybe the protein needs to get cut or, put together with other molecules. That's modification. The Golgi apparatus also can ship. Okay, so it can take those modified products, those protein, and it can bud off the other side. So there's an in to the Golgi and there's an out. So you can see how another vesicle has emerged from the other surface of the Golgi. This guy is going to fuse with the plasma membrane and basically secrete the substances that were made and modified. So it knows how basically to package up all the same molecule and to move it to a very specific place in the cell. So modification and basically transport, like shipping things to different areas of the cell. The last thing here that we see is something called a lysosome. Lysosomes are tiny little, basically membrane-bound sacs. They're very small. And inside the lysosome, we have an acidic interior, and we have a whole bunch of enzymes that degrade molecules. So it's, we call those hydrolytic enzymes. They break down things. So think of this as like the destroying, sort of the stomach, if you will, of the cell. So if something the cell takes in, so let's pretend that this is an immune cell and it got a bacteria, Just because it brought in the bacteria doesn't mean that it's going to kill the bacteria. So the lysosomes come in, they fuse with the bacteria, and then the acid and the enzymes kill it. So it's basically to break down substances inside the cell, lysosomes. And lysosomes are made by the Golgi apparatus. So Golgi makes those guys. All right, so this is just a blow up of the rough ER, okay, studied with ribosomes for synthesis of membrane proteins. So something that goes up to the membrane or secreted out is what the rough ER does. The Golgi body or the Golgi apparatus, I misspelled that, but it is that flattened sacs, a series of flattened sacs. You have an in and an out. So we have transport vesicles from the ER. Don't worry about the cis face or the trans face, but you'll fuse. So these little vesicles full of cargo will fuse to one side of the Golgi, move through the different membranes of the Golgi apparatus, right? And we know that it's getting modified, and we know that it's getting sort of put together. All the like molecules are getting put together, and then when they come out the other side, right, packed up and ready to go, these vesicles are going to be moved to where that cargo should go, mainly a membrane or outside the cell. Okay, so the Golgi apparatus modifies lipids and proteins, producing glycolipids and glycoproteins, and then they are commonly inserted into the plasma membrane. Next up, we have a peroxisome. So this is another just round membrane-bound circle, right? So inside of this circle is different than a lysosome because a peroxisome contains hydrogen peroxide. So hydrogen peroxide is also a dangerous substance. So hydrogen peroxide is H2O2, H2O2, and it has a role in also killing and detoxifying substances that come inside the cell. Okay, eukaryotic structures play a role in breaking down. So I really concentrate on breaking down various molecules. They have a role in lipid biosynthesis, but if we want to just remember breaking down molecules, that's good. All right, next up we have the cytoskeleton. So inside the cell, a eukaryotic cell, we have a series of protein filaments. So think of them as little threads, little like ropes, but they're very sturdy and they create basically the scaffolding inside the cell. So when cells have a particular shape to them, it is the cytoskeleton that makes that shape because remember the plasma membrane is floppy. It doesn't really have a rigid structure to it. So the inside of the cell is that rigidity. Don't think of it so much as our skeleton. Think of it as these guys are really dynamic. They're always, building up or breaking down. Cells can move, like that immune cell that can track bacteria. They can move because the cytoskeleton is rearranging itself inside the cell to push it and to make new shapes. So I'm not going to have you guys know each one of these in terms of like specifics, but you want to recognize the names. So the 3 different cytoskeleton filaments, there's a microfilament, which is the smallest. They have intermediate filaments, which is the next medium size, right? And then we have something called microtubules. And these all work together in different ways for basically anchoring the organelles inside the cell. They can move the cell, but they are plentiful inside the cell. So all these cytoskeleton elements are found in eukaryotic cells. The next organelle here is a centrosome. A centrosome, it looks like 2 penne pastas to me at right angles. These 2 are made of microtubules and they basically function during mitosis and meiosis. So if you learn this in bio, right, we have a centrosome on both ends. When a eukaryotic cell divides, it has to make sure that the DNA that is copied go equally to the new cells. So these guys help make the 2 different ends that you're going to pull the DNA too. So they're very important in basically when the cell divides during mitosis and also in meiosis. Okay, so here they're basically organizing centers of what's called mitotic spindles during mitosis. But just know that they organize, they create these microtubules, which is called spindle fibers, and they grow and they attach to our chromosomes and they pull the chromosomes to one side. All right, we talked about this guy before, a mitochondrion. So remember that this was at one time a free-living prokaryote. So it has things in it that make it autonomous, such as that circular DNA. So we can see the DNA is pictured as circles. It has ribosomes that are different size ribosomes than the rest of the cell. It has a double membrane. It has an inner membrane and it has an outer membrane. For mitochondria, the inner membrane is folded and sort of convoluted. So there's a lot more inner membrane than there is the outer membrane. And that is very on purpose, very on purpose, because we'll talk about how the inner membrane is really important for making energy. So again, the mitochondria is the organelle that is responsible for making most of our ATP. And that's why it's sometimes called the powerhouse of the cell. It makes all the energy that the cell requires. And it's autonomous, so it can duplicate itself if it needs to. If it thinks the cell needs more energy, this mitochondria can just make another mitochondria. So the folding up of that inner membrane has a name. It's called the cristae. So that's something you want to remember. I know it seems like a small detail, but we will talk about that later. And then the inner part here is called the matrix. The chloroplast, this is our other organelle that it was thought to be a prokaryotic organism at one time, right? Again, it has two membranes, inner membrane, outer membrane. It has energy producing, it is not energy, but it's glucose, right? So it can actually make sugar. So that cells make sugar from what, right? Make sugar from carbon dioxide and water, which are both inorganic substances. So this has the ability to use the gas, carbon dioxide, and water to make this organic sugar, which is pretty amazing. And this is how plants can survive with sunlight and water, because they have these organelles that make glucose. Now, like I said, glucose is made here, but then plants also need to break the glucose down to make energy. So plants will have a chloroplast to make sugar, and they will also have mitochondria to make ATP. And we will go into the details of these structures later. All right, the cell membrane of eukaryotes is the same as bacteria. It is a phospholipid bilayer, many proteins embedded in them. It does contain some cholesterol. All right, that's different. Cholesterol is found in all of our membranes. It's a normal part of our membranes. And it is not, if you have high cholesterol, low cholesterol, this is not impacted in any way. So our cells need this cholesterol. And you can, the cholesterol in the membranes help maintain a certain fluidity. Okay. It maintains how fluid the membrane is. So if you think about these phospholipids, they are always in motion. So they're wiggling around, they're moving around, they're not set in place. And they can move quickly or they can move slowly. It is the cholesterol that act as like bumpers or like speed bumps that stop the molecules from moving too quickly, and it maintains a nice fluidity to the membrane. Again, we have glycolipids and we have glycoproteins, right? And largely they act as like recognition of other cells and recognition of pathogens. All right, we're wrapping it up. The process of endocytosis, we already talked about phagocytosis, which is cell eating, but there's other kinds of endocytosis. So endo means to bring in, and cytosis is the cell. So let's look at phagocytosis. This could be the membrane of your immune cell. And these really large bacteria, they're not going to enter the cell through diffusion. They're not going to enter the cell through facilitated diffusion. Because remember, those are tiny. If we have a little protein set up, like the molecules that move through that protein have to be small. So how can you get a really huge particle into the cell? The cell will actually form a divot or an invagination, right? It'll start to surround this large particle. And then as it sort of totally moves around, it will eventually bud off or pinch off. And then you have a vacuole. This term vacuole is different than vesicle, okay? Vacuoles tend to be used for larger things. And this one in particular, like a large bacteria, would be in a vacuole. So it's the same kind of concept where it's just a membrane-bound circle or a sphere, but it's particularly used when you have a large particle inside of it. Pinocytosis, okay, this is cell drinking. So think about Pinot wine. What happens in pinocytosis is that it drinks, right? Cell drinking. So all of these like little sugars or little particles inside the fluid outside the cell, what the cell will do is just take random drinks, random little sips. So it'll make a little invagination of the plasma membrane. It will capture whatever it captures inside the fluid and just brings it in. So it's very non-specific. These two are pretty non-specific in that like anything that it grabs, it can just bring in. So the cell doesn't really know what was in the fluid, but it just brought some fluid in. The last kind of endocytosis here is called receptor-mediated endocytosis. And now we're taking up things that are targeted and very specific substance. And it binds to a receptor. So for receptor-mediated, right, we have a receptor. And receptors are special proteins that are on the membrane, and they fit only a particular kind of molecule. So they're going to target one kind of molecule. In this case, we see it's these beige or these mauve shapes coming in. So these guys are going to attach to the receptor. When the receptor is attached to its ligand, the ligand is a word that is just something that binds to a protein. It will bring it inside of a vesicle. So the cell specifically knows that it's bringing in this substance because it has created receptors to reach out and grab it, right? So it's very specific. So all of these are ways that the cell can move things in through forming a vesicle, through creating that dip or invagination in the plasma membrane, and then pinching off to move the substances inside. The only difference here is that we have a vacuole for the large things. All right, so lastly, we're going to wrap up with prokaryotic versus eukaryotic cells. So this is a nice Venn diagram where we have prokaryotic cells are small and simple. They're largely measured in 0.1 to 5 micrometers. Eukaryotic cells are larger than that. They're 10 to 100 micrometers. So you can kind of see they're almost like 100 times bigger. Prokaryotic cells are only single-celled. So only, remember what had prokaryotic cells? Only the domain bacteria and the domain archaea, right? These are the two groups, A, that had prokaryotic cells. They are life that have these form of cells. Eukaryotic cells are everything else, right? Plants, animals, fungi, protists, they are all eukaryotic cells. So they can be unicellular, like an amoeba or those images of protists that we saw consisting of just one cell, or they can be a multicellular organism like we are, right? Like really large plants, or it doesn't have to be a large plant, but any plant or animal. The nucleus is absent because they have a nucleoid, and it's a circular kind of DNA inside their nucleoid. We, eukaryotic cells, have a nucleus, and the DNA that's found is linear. So it's a line or a straight, straight molecule. Let's not worry about haploid and diploid chromosomes. This is really important, right? We do not have membrane-bound organelles inside prokaryotic cells. We have membrane-bound in eukaryotic, these guys reproduce sexually and asexually. Most of the plants and animals and fungi, they produce sexually, not all, because again, eukaryotic cells also include protists, and some fungi produce asexually. Cell division. Oh, so let's talk about sexually here for bacteria. Sexually means that you're basically just mixing DNA, right? It doesn't mean that these bacteria, quote unquote, have sex, but sexually means that you're going to have genetically different offspring than the parent. And then asexually means that your offspring are exactly like you, right? You're making a clone. So we'll talk about how some bacteria can actually mix up their DNA and have sexual reproduction. The division of bacteria is a simple process called binary fission. The division by eukaryotic cells when they divide is more complicated and is called mitosis. I did not ask you guys to learn mitosis, but you should recognize it as something that eukaryotic cells do. And then examples are bacteria and archaea, examples plant, animal, and humans, of course. How are they similar? They all have a cell membrane on the outside. They all have cytoplasm. They all have ribosomes, and they all have DNA. All right, some questions at the end. What is the main structural feature that eukaryotic cells possess but prokaryotic cells lack? All right, and we should recognize that the nucleus is our eukaryotic feature. Which of the answers below describes one of the functions of the bacterial cell wall? Okay, so if you look at these, the only one that makes sense here is going to be osmotic pressures, because it does not allow the cell to lyse or burst open. It does not protect from antibiotics. It does not protect against temperature extremes, and does not protect from drying out. The drying out is your capsules and your slime layers. Active transport across the cytoplasmic membrane differs from facilitated diffusion in what respect? So we have active transport and we have facilitated diffusion. The answer is B. Active transport is used to move materials against their electrochemical gradient. Electrochemical just means that some things have a charge, but it moves against their gradient. So you're not moving from high concentration to low concentration. You're actually moving from low concentration to high concentration. And then, yeah, so this answer D, only active transport uses transport proteins, is false, because for facilitated diffusion, you have to facilitate those molecules to fuse through proteins, right? So through those transport or channel proteins we mentioned. And that's it.